You know, when you usually think about a medical diagnosis, there's this certain expectation of precision. It feels almost like engineering, right?
Yeah, exactly. It's very mechanical.
Right. Like you break your arm, the x-ray shows that stark, jagged white line across the bone, and the doctor just points at it. There it is.
Broken or not broken?
Broken or not. It's clean, it's visible, and honestly, it's pretty comforting to have that clarity.
Well, it is comforting because we are kind of hardwired to, like, things we can clearly categorize. But when you step into the world of oncology, and specifically, you know, the tumor microenvironment, that X-ray machine is utterly useless.
Yeah, I mean, it's just completely murky.
We are looking at a biological landscape that is incredibly murky and chaotic.
Murky is definitely the perfect word for it. So imagine your liver for a second. It is not just this static organ sitting in your abdomen. You know?
Right, it's highly dynamic.
Exactly. It's this bustling, highly regulated, microscopic city. You've got complex traffic control, intense waste management, and a local cellular police force keeping everything in check.
But then a stealthy enemy moves in.
Yes, and it doesn't just hide. It systematically corrupts that local police force, so they actually start protecting the invaders.
Which is the absolute definition of diagnostic and therapeutic muddy waters. The cancer turns the city's own biological defenses against it.
Which brings us to our mission for today's deep dive. We are looking at a truly groundbreaking 2026 paper published in the journal Cell Death and Disease.
It's a fantastic piece of research.
It really is. The focus here is hepatocellular carcinoma, or HCC, which is the most common and honestly notoriously difficult to treat form of liver cancer.
And our goal today is to explore every conceivable detail of how a completely surprising biological structure is involved here.
Right. A simple cellular salt channel, one that is usually associated with the lung disease cystic fibrosis.
Exactly. And we're looking at how that tiny channel can radically reprogram the immune system to fight off this liver cancer. It is a massive paradigm shift.
A total shift. So we are going to trace the exact molecular domino effect today, from start to finish, explaining every single term.
Down to the specific ions and electrical charges. And we'll dive into the rigorous testing that proves this isn't just, you know, some interesting theory.
Right. It's a real potential turning point in cancer immunotherapy. So let's jump right into the biology. To understand how to win this fight, we first have to understand the battlefield itself, right?
The immune microenvironment of HCC.
Yeah. So it helps to know the cast of characters here. First, you have your CD8 positive T cells.
In the cellular world, these are the undisputed good guys.
The assassins.
Right, they are cytotoxic lymphocytes, meaning their entire biological purpose is to patrol the body, identify aberrant or cancerous cells, and just assassinate them.
They are the elite strike force. I mean, if they are in the neighborhood, cancer usually loses.
True, but in HCC, this strike force is completely shut out. They are outnumbered by the second character we need to talk about.
Regulatory T cells.
Or TREGs for short. In a healthy body, TREGs act like the bouncers of the immune system.
They keep things calm, right?
Exactly. They secrete dampening signals to calm things down so your immune system doesn't accidentally attack your own healthy tissue.
Which is what causes autoimmune diseases.
Right. But the tumor hijacks them completely. In the HCC microenvironment, TREGs can account for up to 30% of the immune cells present.
Wow, 30%. That's huge.
It's massive. They throw this protective blanket over the tumor, actively suppressing those CD8 positive assassins.
Okay, so that covers the assassins and the bouncers. But the paper spends a lot of time on a third group, which are the macrophages.
Macrophages are fascinating because they are basically the shapeshifters of your immune system. Depending on the chemical signals around them, they polarize into two main states.
Right, the M1 and M2 states?
Yes. M1 macrophages are the classical pro-inflammatory cops. They fight tumors, they secrete tumor-killing toxins, and they sound the alarm for backup.
And then there's the alternative.
M2 macrophages. These are essentially corrupt cops. When they get polarized into the M2 state by a tumor, they stop fighting entirely.
They just switch sides.
They do. They start promoting tumor growth and facilitate a process called angiogenesis.
Which is the creation of new blood vessels, right?
Exactly. A growing tumor is starving for nutrients, and M2 macrophages help build the supply lines directly to it. Plus, they actively suppress any lingering immune response.
So if I'm mapping this out, this completely explains why our current best weapons in oncology are failing.
It really does.
Because right now, first-line treatments for HCC include immune checkpoint inhibitors. We're talking about PD-1 or PD-L1 monoclonal antibodies.
Drugs like nivolumab or atezolizumab.
Yeah, but the paper points out that the response rate for these incredible drugs is below 30%.
Because HCC is what oncologists call a cold tumor.
A cold tumor, meaning it's biologically desolate when it comes to the good guys.
Precisely. The environment lacks CD8 positive T cells, but it is absolutely overflowing with those suppressive tregs and corrupt M2 macrophages.
It's like, okay, using a PD-L1 inhibitor on a cold tumor feels a bit like unlocking the front door of a massive fortress to let a liberating army inside.
I like that analogy.
Yeah. The key works perfectly, right? The door unlocks. But if there is no army standing outside, meaning no CD8 cells, and the inside of the fortress is packed with enemy guards like tregs and M2s, unlocking that door accomplishes absolutely nothing.
Nobody's coming to the rescue. The drug works mechanically, but the local environment is just entirely hostile to its success.
You cannot just unlock the door. You have to fundamentally change the neighborhood.
Exactly.
So let's do a quick recap of this first part. The environment of this cancer is cold, meaning it's packed with bad guys and the good guys are locked out, which is why standard miracle drugs fail.
That's the baseline problem, yes.
So have you ever wondered why miracle cancer drugs only work for a fraction of patients? The secret is in the neighborhood, not just the tumor.
Which leads to the big question of how we change that neighborhood.
Right, how do we evict these bad cells? The paper points to a completely unexpected culprit.
Enter CFTR, the cystic fibrosis transmembrane conductance regulator.
CFTR. I mean, when I hear that, I immediately think of cystic fibrosis, the genetic lung disease.
Most people do.
What is a lung disease protein doing in a liver tumor?
Well, it turns out CFTR is fundamental to many parts of the body. It's a massive, complex protein made of 1,480 amino acids.
All right, that's huge.
Structurally, it acts as a chloride ion channel. Picture a microscopic pipe embedded directly in the membrane of epithelial cells.
Like the hepatocytes in your liver.
Exactly, and the cells lining your bile ducts. Its normal day job is to transport negatively charged chloride ions in and out of the cell.
Which regulates the movement of water and salts.
Right. In the liver, this pipe is vital for healthy bile secretion.
So how did the researchers connect this microscopic salt pipe to a deadly liver cancer?
They actually didn't start with a hypothesis about salt at all. They started by analyzing massive open-source genomic and transcriptomic databases.
Like TCGA, GPI, and G2Hex.
Yes. By running algorithms across hundreds of patient tissue samples, they were looking for any gene expression that was drastically different in cancer versus healthy liver tissue.
And the data on CFTR was startling, right?
It was. CFTR is massively downregulated, essentially silenced in HCC tissues.
Wait, when you say silenced, is that a genetic mutation? Like the patient is born with a broken CFTR gene, similar to cystic fibrosis patients?
No, and that's the crucial part. It goes deeper than that. It is not a structural mutation.
Oh, really?
The researchers checked the mutation rate of the CFTR gene in these HCC patients, and it was a tiny 1.35%.
So the genetic blueprint for the pipe is completely intact?
Completely intact. But the cancer environment is actively suppressing the manufacturing of it.
And the clinical reality for patients is pretty stark. The lower your CFTR expression, the worse your prognosis.
Right. More advanced tumor stage and poorer cellular differentiation.
Wait, let's unpack this for a second because I want to push back. You're talking about a microscopic salt pipe.
A very tiny pipe, yes.
How on earth does a plumbing issue, just moving chloride ions back and forth, dictate whether a cancer becomes aggressive or how immune cells behave?
It sounds absurd, I know.
It feels like saying the plumbing layout of a house dictates the political beliefs of the people living inside it.
That's a great way to put it. But in biology, plumbing actually dictates the fundamental physics of the cell.
The physics?
Yes. The researchers used a cutting-edge technique called spatial transcriptomics to prove this.
And spatial transcriptomics, just so we're clear, doesn't just tell you what genes are active in a blender full of tissue, right?
Exactly. It creates a highly detailed physical map, showing exactly which genes are turned on at exact microscopic coordinates on a tissue slice.
It's like biological GPS.
They mapped it out and proved that CFTR is physically missing from the outer boundaries of the tumor cells.
And right where that pipe is missing, the aggressive immune suppression is most intense.
Exactly.
So just to recap this section really quickly, CFTR is this hard-working salt channel, but in HCC, it mysteriously vanishes, not from a mutation, but because the tumor actively silences it.
And that silencing leads to deadly cancer.
So, can a microscopic salt channel really act as a master switch for the entire immune system? To answer that, we have to look at the exact mechanism.
We have to trace the molecular domino effect. This is the dense, beautiful biochemistry of how a missing pipe alters cellular physics.
I am ready. Let's take this step by step. What is the very first domino?
Domino 1 is ion buildup. When the CFTR pipe is lost or silenced, chloride ions, which carry a negative electrical charge, cannot escape the tumor cell.
So they just get trapped in there.
They get trapped and build up inside. This accumulation of negative charge dramatically alters the cell's membrane potential.
Membrane potential, meaning the electrical voltage across the cell wall.
Exactly. Cells operate like tiny batteries. When that electrical voltage shifts violently due to the trapped chloride, it triggers a secondary disaster.
Ages what?
The voltage change forces open other channels on the cell surface, leading to an abnormal, massive influx of intracellular calcium.
So the plumbing is blocked, chloride backs up, it changes the electrical charge of the cell, and that electrical panic drags a flood of calcium into the mess.
Exactly right.
What happens to all that trapped chloride and calcium?
That triggers domino 2, which involves the transcription factors. These high levels of chloride and calcium don't just sit around aimlessly.
They act as messengers, right?
Urgent messengers that travel deep into the nucleus of the cell. Their job is to wake up two specific transcription factors.
Okay, let's define transcription factors for a second for the listener. These are proteins that float around in a dormant state.
Right, they are inactive at first.
But when activated, they physically bind to specific sequences of your DNA to turn genes on or off, sort of like a light switch.
That's spot on. The two transcription factors awakened here are RUNX1, which responds directly to the chloride imbalance.
RUNX1, got it.
And NF-κB, which is a classic responder to calcium flooding. They bind to the promoter regions of the DNA and flip the switch.
Okay, so they bind to the DNA. What exactly are they switching on?
That is domino 3, the cytokine SOS.
Yeah, SOS.
Yes. Once RUNX1 and NF-κB bind to the DNA, they instruct the tumor cell's machinery to frantically manufacture and pump out specific signaling proteins.
Cytokines and chemokines.
Yes. The researchers pinpointed 4 main culprits being blasted out of the cell: CSF1, TGF-beta, CCL20, and CCL22.
Let me remember that. CSF1, TGF-beta, CCL20, and CCL22. We could call these the corrupt cop calling codes.
Corrupt cop calling codes. I love that. They are basically acting like corrupt dispatchers, actively radioing for the exact wrong kind of backup.
Which brings us to the final domino, right? The immune shift.
Exactly. These 4 chemical dispatch signals leak out of the tumor cell and into the surrounding microenvironment.
And what do they do when they get out there?
Well, TGF-beta and CSF1 are highly specific signals. They find normal, resting macrophages and corrupt them, forcefully polarizing them into that bad M2 state we talked about earlier.
So they literally turn good cops bad.
Yes. Meanwhile, CCL20 and CCL22 act as powerful chemical magnets.
To recruit the bouncers.
Exactly. They specifically recruit those bouncer trags out of the bloodstream and directly into the tumor.
It is incredibly sinister when you lay it out like that. By simply losing a single salt pipe, the cell floods with negative ions, suffers an electrical panic, turns on genetic switches in the nucleus, and blasts out a chemical radio signal that builds the cold tumor environment.
It is a brilliant, terrible system.
But if that mechanism is true, it implies a pretty massive therapeutic vulnerability, doesn't it? Right. What happens if you just force the pipe open again?
That is the major therapeutic breakthrough of the paper. The researchers proved that if you force the cell to overexpress the CFTR gene using genetic tools, you can fix it.
Or even better, using drugs, right?
Yes, if you use specific channel-opening drugs like forskolin or VX770.
VX770 is an already approved cystic fibrosis drug, right?
It is, and it physically forces the remaining pipes to open wide.
So imagine the tumor cell as a sealed room. There's a drain on the floor, that's your CFTR channel. If the drain gets maliciously plugged, the room starts rapidly filling up with water, the chloride and the calcium.
The flooding.
And that flooding triggers a panic alarm on the wall, RUNX1 and NF-κB, and that deafening alarm calls in all the bad guys to surround the building.
But if you simply unplug the drain with a drug like forskolin,
the water rushes out, the room normalizes, the alarm shuts off, and the bad guys disperse.
The elegance of it is truly stunning. When they applied the drugs, the chloride and calcium levels dropped right back to baseline.
And the transcription factors.
RUNX1 and NF-κB detached from the DNA and just went back to sleep. The corrupt dispatcher signals completely stopped broadcasting.
And without those chemical magnets, the microenvironment just rapidly flipped.
The corrupt M2 macrophages vanished, and M1 macrophages, the good cops, along with CD8 positive T cells, flooded back into the tumor.
Okay, time for a quick recap of this dense section. We just traced this incredible biological cascade. CFTR loss traps chloride,
Which pulls in calcium,
which wakes up RUNX1 and NF-κB in the nucleus.
which secrete the cytokines that build the biological fortress.
And fixing the channel reverses the entire chain. So ask yourself, are you starting to see how manipulating invisible microscopic ions fundamentally changes the biology of an entire organ?
It really is all connected.
But here's the thing. In science, a beautiful logical mechanism is absolutely nothing without ironclad proof.
Oh, definitely. You cannot just claim you slip the immune system in a petri dish and call it a day.
So how did the researchers prove this? They must have thrown the absolute kitchen sink at it in vivo.
They did. But first, let's look at how they even measured the chloride.
Right. How do you measure invisible chloride ions inside a microscopic cell?
They used a brilliant technique involving MQAE fluorescence.
MQAE fluorescence.
Yeah, MQAE is a special chemical dye that you introduce to the cells. It has this unique property where it fluoresces or glows brightly normally, but its glow is quenched, meaning it gets dimmer the more it collides with chloride ions.
Oh, that's clever.
Right. By placing these cells under a highly specialized microscope, they could visually measure the chloride buildup in real time just by tracking how dark the cells became.
Okay, so they proved the chloride backs up visually. But how did they prove the tumor cells were actually corrupting the immune cells?
For that, they set up a co-culture system. They took human HCC tumor cells and placed them in a dish with raw 264.7 cells.
And those are a standard line of resting macrophages, right?
Yes, but they separated them with a permeable membrane, meaning the cells couldn't touch each other physically, but the chemical dispatch signals could float across.
And what happened?
They literally watched the resting macrophages polarize into the bad M2 state based solely on the chemical cytokines the tumor cells were secreting.
Wow. That handles the isolated cells. But obviously, biology in a dish is very different from a living, breathing body with a complex immune system.
Which is where their animal models were so meticulously designed, you can't just jump to a normal mouse.
Why not?
First, they used immunocompromised mice strains known as BLB/CA nude and NOD/SCID mice.
These mice lack functioning immune systems entirely.
Right. This allowed the researchers to observe how CFTR changes affected the raw growth of the tumor itself without the immune system interfering at all.
Oh, so they were establishing a baseline.
Exactly. Once they knew the baseline, they moved to immunocompetent mice, C57BL/6J mice.
Which have fully functioning immune systems?
Yes. They implanted tumors subcutaneously under the skin, but more importantly, they implanted them orthotopically.
Meaning directly into the mouse's liver.
Which is surgically difficult, but it provides a much more accurate mimic of true human HCC.
They also grew human HCC organoids, didn't they?
They did.
And organoids are just fascinating. They take an actual biopsy from a human cancer patient and grow it into a miniature 3D tumor structure in the lab.
It behaves much more like real human tissue than flat cells in a dish ever could.
It's basically the gold standard for testing short of a human trial.
It is.
Yeah.
And this set the stage for the ultimate climax of the paper, the synergy test.
This goes all the way back to our locked fortress analogy with the PD-L1 immunotherapy drugs.
Precisely. When they treated the mice with just a PD-L1 antibody, the results were modest at best.
The drug unlocked the door, but just like in humans, the response was poor because the tumor was still cold.
But when they combined the PD-L1 antibody with the CFTR activator drug for scolin,
the data was staggering.
Beyond staggering. The neighborhood changed completely. The tumor went from cold to hot.
CD8 positive T cells massively infiltrated the tumor tissue.
The corrupt M2 macrophages were replaced by aggressive M1 macrophages.
The actual physical weight of the tumors plummeted.
And the rate of metastasis, the cancer spreading to other organs, dropped significantly.
By simply opening a salt channel, they completely removed the tumor's biological shield, and the standard immunotherapy drug suddenly worked with devastating efficiency.
It's beautiful to see that kind of synergy.
There's a detail from the single-cell RNA sequencing data in this paper that absolutely blows my mind.
Which part?
The researchers proved that the CFTR salt channel is only found on the epithelial tumor cells.
Right.
It does not exist on the immune cells themselves, not the T cells, not the macrophages.
Not at all.
Yet, by purely fixing the internal chemistry of the tumor cell, they achieved complete control over the immune cells operating outside of it.
It is like the tumor cell is a puppeteer.
And the chloride and calcium ions are the strings.
It really speaks to the deep interconnectedness of the tumor microenvironment.
Yep.
We have spent decades trying to brute force the immune system with increasingly toxic drugs.
Yeah.
This paper proves that sometimes you don't need to drug the immune system at all. You just need to strip the tumor of its ability to manipulate its neighbors.
It is brilliant. So let's do a quick... is that dim in the presence of chloride, they observed real-time macrophage corruption across permeable membranes.
And they used layered mouse models, from immunocompromised to fully competent,
to prove that combining a simple salt channel activator with standard immunotherapy creates a massive tumor-destroying synergy.
Ironclad proof.
So consider this. What if the key to unlocking the future of cancer immunotherapy isn't just creating more complex engineered immune cells, but simply reminding the tumor cells how to open a drain?
It forces the entire oncology field to take a step back. We spend billions looking for complex, rare genetic mutations to target with precision medicine.
But this shows us that sometimes the most deadly weapon a cancer has is a fundamental disruption in basic, ancient cellular housekeeping.
Like the balance of salt and water.
So let's bring this all together for our final synthesis. We started this journey looking at hepatocellular carcinoma, a deadly, notoriously untreatable liver cancer.
That survives by hiding behind a biological fortress wall built out of corrupt macrophages and suppressive T cells.
But through the incredibly detailed molecular detective work in this 2026 paper, we discovered that this fortress isn't built with bricks or unbreakable genetic mutations.
It is built with blocked plumbing.
By forcing open a single cellular drain, the CFTR channel, we can stop the electrical panic inside the cell.
Flush out the toxic chemical dispatch signals.
Clear out the bad actors in the microenvironment and let life-saving immunotherapies finally do the job they were designed for.
It is a stunning victory for basic biological science. By persistently asking why a cancer cell has altered its salt levels, these researchers may have unlocked a synergistic treatment pathway that could eventually save countless lives.
Which leaves you with a final, mind-expanding question to mull over as you go about your day.
Let's hear it.
If an older drug designed to treat cystic fibrosis, or a naturally occurring compound like forskolin, can fundamentally rewire the immune system of an aggressive liver tumor just by moving microscopic salt ions around.
What else is out there?
Right. What other forgotten everyday ion channels scattered across our billions of cells are secretly dictating the fragile line between health and disease?
It really makes you rethink every complex disease out there. The answers might be hiding in the simplest mechanisms.
It absolutely does. Thank you for taking this highly detailed, wonderfully complex journey into the tumor microenvironment with us on today's deep dive. Keep questioning the invisible biological mechanisms operating inside you every single second. And remember, sometimes the biggest, most terrifying mysteries can be solved by simply fixing the plumbing.